Hi, my name's Dr. Hilda High. I'm a genetic oncologist, a cancer specialist working in hereditary cancer syndromes. This is just a brief video on informed consent. In other words, what do you need to know in terms of the implications and limitations of a genetic test? We do have more detailed information in terms of videos on our website, particularly if you have a personal or family history of breast or ovarian cancer, or maybe your family history is more about polyps and bowel cancer. There's also information on interpreting genetic test results and links to information on how to manage hereditary cancer risk. In terms of this video, though, it's about informed consent. So let's get started. Well, it helps to know that most cancer is not caused by inherited mistakes. Instead, cancer is caused when mistakes build up in the DNA of cells. If genes that tell cells when to grow, when to die, and how to behave are damaged as that cell is dividing and growing, then you'll end up with a cell that grows when it shouldn't, doesn't die when it should, and stops following the rules. That's a cancer. But those mistakes, those mutations have just occurred in that particular cell, just in the DNA of that cell. They're not inherited and they're not passed on. But in some families, there are inherited mistakes. What's that about? Well, a gene is a code, a recipe. Most of our genes make a protein, and the protein then does something. So if that code, that recipe isn't working, then either the protein won't be made at all, or it won't be able to do its job. So I always use the example of a cake recipe. Beat eggs well. Now you probably know the DNA code is in groups of 3, not 4, but it's the same idea. If I'm writing this out, I have to keep the pattern working. So adding in or leaving out one letter will be enough to ruin the whole thing. If I change one of those 3-letter codes in the DNA, or in this case, one of the 4-letter words in the recipe, it'll depend on what I change and what I change it to. So beat eggs good, not very good English, but the cake will probably work, whereas beat cats well is not going to make a good cake. So all the genetic test does is look at the code of the gene to see if there are any changes that could cause trouble. When we find a change, just like with a cake recipe that you could ask a talented chef to interpret, too wet, too dry, too many eggs, we have protein prediction tools that will tell us whether this particular change is going to cause problems. These changes are then divided into 3 different groups. Those that do cause trouble, these are the class 4 and 5 variants, those that we don't yet understand, and if we don't understand it, we really just don't understand it. We put it to one side and we try and sort this out later. And then there are changes that we all carry, but we don't think they cause us trouble. We call these polymorphisms if they're common. We all carry these changes, and luckily the labs won't tell us if they're there. If it doesn't cause you trouble, they're not going to tell you about it. So how do these mutations, these pathogenic variants in a cell cause trouble? Well, if you've inherited a mistake, if it came over in the egg or in the sperm that made you, it was there when you were 1 cell, 2 cells, 4 cells, 8 cells, it's going to end up in every cell. But you'll still have a second copy. And that's because most of these cancer risk genes don't sit on the sex chromosomes. So you'd know you get 2 copies of every gene, one from mum in the egg, one from dad in the sperm, except for those on the sex chromosomes. Women do still have the 2 X's, but don't have a Y, men an X and a Y. But for everything else, there are 2 chromosomes, so you get 2 copies of every gene. The genes that tend to cause trouble in cancer perform 2 key roles. The first group you can think of as spell checker genes. Their job is to look for and fix the mistakes that build up in the DNA when cells are dividing. If you inherit a mistake in a spell checker gene, it was there when you were 1 cell and it'll be in all the cells, but you'll still have a second working copy, at least for most of these genes. And that's because they're just too important. For example, with the BRCA genes you might have heard about, they protect us against breast, ovarian, prostate, and pancreas cancers. This gene is just too important. You can't have two mistakes in the BRCA genes. Either the baby won't grow at all, that's usually the case with the BRCA1 gene, or they end up with a devastating condition. In the case of BRCA2, that's normally Fanconi's anemia. We know about that when they're very young. So for spell checker genes, while you may inherit one mistake, the second copy is normally working. And that's good news, because that second copy, it's going to be in every cell as well, and it can protect you. The problem is, we just said any gene can be damaged, switched off, or interfered with. If that happens to the second copy, that particular cell has now lost both recipes. No gene, no protein, no spell checker. So it's not that you make any more mistakes, it's just the mistakes we're all making can build up faster in that particular cell, and that's why we start to see more cancer and cancer at a younger age. You can probably guess how the growth genes work. Here, you've inherited a mistake in a gene that's normally left switched off. That's going to cause problems often from a younger age, and growth genes often go with outward signs, maybe particular types of lumps on the skin, birthmarks, or other problems elsewhere. These are usually things we know about when somebody's quite young. So two forms of genes cause problems: spell checker genes and growth genes. If you've inherited a mistake, it's going to end up in every cell. And that's why when we do our genetic testing, we don't have to look anywhere tricky. Any cell with some DNA will do. The DNA is inside the nucleus. White cells have a nucleus, so most genetic testing in Australia is done via a blood test. There are white cells in saliva. You can use cheek or gum scrapings, even the hair bulbs. For most testing, though, as we said, we just use blood. It doesn't matter where the DNA has come from, so long as it's representative of the whole of your body. Once we have the DNA, then we're just going to check the copy from mum and the copy from dad. We're not trying to tell which one's which. It's not that kind of test. We're just looking for one of those spelling mistakes or pieces missing. Now, if we did find something, that's the easy part. That genetic change, that mutation, it's been there your whole life. Now that we know about it, we can do something about it. And once we know which book, which chapter, which word, then we can offer the test to another blood relative. In fact, you may be having your testing looking for a mistake that we already know about in a blood relative. That kind of testing is called predictive testing because it's very yes, you carry that same change, or no, you don't. So a predictive test is a very yes or no type of test. If we do the test in someone and we don't find a mistake, well, we can't always go, oh, well, don't worry, we haven't explained the cancers in a family. So it's important to realize that a genetic test can only tell you about risk. Not finding a mistake, we call that uninformative. Uninformative meaning genes are working, didn't explain the cancers. For some families, your doctor may still recommend more screening or may refer you to a research study or a specialist cancer centre. So where do these mistakes, these pathogenic variants or mutations come from? Well, somebody has to go first. We call this a de novo mutation. For the spell checker genes, this is not common. For the growth genes, it may be more common, maybe 30% of the time that may have been what happened. The egg or sperm that made you was working fine, but as you went from 1 cell to 2 cells to 4 cells to perhaps 8 cells, a mistake was made. That's going to end up in all of your cells, and that means it can also be passed on. On the other hand, if you inherit a mistake, if it's there when you're 1 cell, it will be in all your cells, and that means it'll be in the cells you use when you make an egg or make a sperm. At that point, you have to split the pair. And that's why for these sorts of genes, where one mistake's enough to put you at risk, it will be 50-50 for each child. 50-50 that they have inherited that genetic change, 50-50 that they haven't. As we said, usually this has nothing to do with the sex chromosomes, so boy, girl, it doesn't matter. This also means if there's no mistake, then it doesn't matter how many eggs, sperm, or babies you make, you can't pass on a mistake that's not there. And also, it explains why we can't go backwards. We can't say no mistake in you means there was no mistake in your mum or dad. They still have two copies. We didn't test the second copy. We just know that the version you inherited, the versions that you could pass on, are working fine. Now, people often worry about insurance. This gets a lot of media play, but in Australia, the insurers have agreed that they won't ask about genetic testing and you don't have to tell. That's for $1,000 a week of income protection, $500,000 of life insurance. It's what people sort of accidentally get through their superannuation. Importantly, this sort of testing doesn't affect travel or health or any existing policy. That's not to say, though, that the cancers in the family don't have an impact. They usually have a much bigger impact. So in terms of insurance, I'm not a financial advisor. Some people do want to get this sorted before they have their testing. Importantly, though, if there is a known mistake in a family member and you don't carry it, you usually go back to being average risk. So in terms of informed consent, the reason it's important is that you need to understand the implications and limitations of genetic testing. We've already run through how this test still won't be able to tell you if you will get cancer, won't get cancer, the age or type. It can only tell you about your level of risk. However, if you are at risk, high risk, there are a lot of things you can do to reduce that risk. Most people go into more detail about that after the results come back. As we've mentioned, because of how these are passed on, your result can have implications for other family members, and in some small situations, it may even reveal non-maternity or non-paternity. We just mentioned the insurance, but I think it's also important to point out if you're having testing via a large panel, then the chance of finding a change that we don't yet understand will be increased. We call these variants of uncertain clinical significance. We just put them to one side, and over time we'll get more evidence and we may be able to reclassify these changes. Sometimes, though, we find a change in a gene that didn't really fit with your family history. For example, we often test the Lynch syndrome genes at the same time as we're testing the breast cancer risk genes. There may be no history of Lynch syndrome-like cancers, bowel cancer, uterine and ovarian cancer, for example, and it may come as a bit of a shock. We call these incidental findings. Generally speaking, though, if there is a high-risk gene mutation, the sooner we find it, the better. Plus, that change, it was there your whole life. Knowing about it means we can do something about it. In terms of the DNA, in Australia, the DNA is stored by the lab that does the test. They don't on-sell it or use the data in any other way except for your clinical result and potentially for the health of the family by providing a control when predictive testing's done. So generally, at the time that you give consent for your genetic test, you'll usually be asked for permission to share the information, not necessarily your personal information, but the information about the mistake that's been found so that other family members can have testing. And of course, with any kind of test, your doctor should run through cost. Testing in Australia is funded under Medicare, that is bulk billed, but only in certain circumstances. For a complete list, have a look on our website or talk to your doctor. So, to summarize, you should now understand that all cancer is genetic. It's all caused by mistakes that build up in the DNA of cells. Only about 5% of most cancers have been caused by an inherited mistake. The rest are just because of mistakes that are built up gradually over time in that cell over that person's lifetime. But for those people where that is a possibility, having a genetic test will allow us to identify a mistake that we can then do something about. As we've also discussed, though, not finding a mutation doesn't mean no risk. So after the test, your doctor will still provide you with some more personalised screening advice. For those where a mutation is found, the EVQ guidelines are the National Management and Testing Guidelines in Australia, and they list gene by gene what kind of risk is present and what to do about it. There is also more information on our website.